Life is a miracle, a complex tapestry woven from countless threads. At the heart of this tapestry lie genes, the blueprints of life itself, dictating everything from eye color to disease susceptibility. Understanding genes is crucial to understanding who we are. In The Gene and Intimate History, Siddhartha Mukherjee unravels the history and profound implications of genetic discoveries. In this video, we will explore the gene, tracing the history of genetic discoveries. From Mendel's pea plants to modern gene editing, we'll meet the brilliant minds who shaped our understanding of genes. We'll delve into the impact of genes on our lives and the ethical questions they raise. Join us on this captivating journey into the world of the gene and intimate history. Our story begins in a quiet monastery garden in 19th century Austria. Gregor Mendel's experiments with pea plants laid the foundation for genetics. He discovered that traits are inherited in distinct patterns, not blended. Mendel's work revealed that traits are inherited as discrete units, later known as genes. Mendel formulated the fundamental laws of inheritance. The law of segregation states that each individual carries 2 copies of each gene, which separate during sex cell formation. The law of independent assortment states that the inheritance of one trait is independent of others. Mendel's groundbreaking work was initially met with indifference, but later recognized as revolutionary. At the dawn of the 20th century, Mendel's work was rediscovered by 3 scientists. They recognized the significance of his findings, providing the foundation for genetics. The rediscovery marked a turning point in biology. Mendel's laws paved the way for groundbreaking discoveries in genetics. The rediscovery of Mendel's work ignited research into the physical basis of inheritance. Scientists sought to understand the hereditary material, DNA. In the 1950s, Watson and Crick, using X-ray diffraction images, discovered the double helix structure of DNA. This breakthrough revealed how genetic information is stored and passed down. The double helix structure suggested a mechanism for genetic information storage and replication. Complementary base pairing ensures accurate DNA replication. The sequence of DNA bases forms a code specifying amino acids and proteins. The genetic code is universal, highlighting the unity of life on Earth. The discovery of the double helix ushered in a new era of biological research. The Human Genome Project, launched in 1990, aimed to sequence the entire human genome. Completed in 2003, it provided an unprecedented view of our genetic blueprint. This knowledge is revolutionizing personalized medicine and various research fields. The human genome is a dynamic symphony score, guiding cell development and function. Genes act as musical phrases, carrying instructions for building proteins. The precise orchestration of gene expression is essential for life. This regulation is a delicate dance between genetic and environmental factors. The journey from gene to trait is mediated by gene expression. DNA is transcribed into mRNA, which is then translated into proteins. Ribosomes read the mRNA sequence and assemble amino acid chains. The dynamic regulation of gene expression allows cells to respond to changing conditions. Errors in genes can lead to a range of health problems. Mutations can arise spontaneously or be induced by environmental factors. Genetic disorders can be single gene, chromosomal, or complex. Single gene disorders follow Mendelian inheritance patterns. Unmasking single gene disorders. Single gene disorders arise from mutations within a single gene. They follow predictable inheritance patterns based on Mendelian genetics. Examples include cystic fibrosis, sickle cell anemia, and Huntington's disease. Each disorder results from a specific gene mutation disrupting protein function. When chromosomes falter. Navigating chromosomal disorders. Chromosomal disorders involve alterations in the number or structure of chromosomes. Down syndrome is caused by an extra copy of chromosome 21. Structural abnormalities include deletions, duplications, inversions, or translocations. Cri-du-chat syndrome is caused by a deletion on chromosome 5. Chromosomal disorders can arise spontaneously or be inherited. The risk increases with maternal age, especially after 35. Section 3. The complexity of inheritance. Deciphering multifactorial diseases. While single gene and chromosomal disorders provide clear examples of how genetic alterations can lead to disease, many common diseases, such as heart disease, cancer, diabetes, and mental illnesses, arise from a complex interplay of genetic and environmental factors. These multifactorial diseases, as they are called, do not follow simple Mendelian inheritance patterns. Instead, they are influenced by variations in multiple genes, often interacting with environmental factors such as diet, lifestyle, exposure to toxins, and infectious agents. Each genetic variation may contribute only a small increase in risk, but the combined effect of multiple risk variants, coupled with environmental exposures, can significantly increase an individual's susceptibility to disease. Unraveling the complex interplay of genetic and environmental factors in multifactorial diseases is a major challenge for researchers. Genome-wide association studies, or GWS, which involve scanning the genomes of thousands or even millions of individuals to identify genetic variations associated with disease risk, have proven to be valuable tools in this endeavor. GWAS have identified hundreds of genetic variants associated with an increased risk of various complex diseases. These findings are providing insights into the biological pathways involved in disease development and may eventually lead to new strategies for prevention, diagnosis, and treatment. Untangling nature's threads. For centuries, philosophers and scientists have debated, what makes us who we are? Are we products of our genes or shaped by our experiences? The answer lies somewhere in between. Our genes play a role in shaping our behavior. But genes are not destiny. They provide a framework for our experiences. Our experiences weave those threads into the tapestry of our behavior. Section 2. Beyond the blueprint. The interplay of genes and experience. One of the most compelling examples of the interplay between genes and environment can be found in the study of identical twins. Identical twins share 100% of their DNA, making them ideal subjects for studying the relative contributions of genes and environment to various traits. Studies of identical twins raised apart have revealed fascinating insights into the complex dance between nature and nurture. While identical twins share remarkable similarities in many traits, from physical appearance to personality quirks, they also exhibit striking differences, particularly in traits influenced by experience, such as values, beliefs, and life choices. These differences highlight the profound impact of environment on shaping who we are. Consider the case of Jim Lewis and Jim Springer, identical twins separated at birth and reunited at the age of 39. The Jim twins, as they became known, shared an uncanny number of similarities, from their taste in beer to their habit of biting their fingernails. However, they also had distinct personalities and life experiences, reflecting the influence of their different upbringings. The Jim twins story, along with countless other twin studies, underscores the importance of both genes and environment in shaping who we are. Our genes provide the raw materials, the building blocks of our being, but it's our experiences that mold those materials into the unique individuals we become. Rewriting the code of life. The advent of gene editing. The discovery of the double helix structure of DNA in 1953 marked a turning point in our understanding of life's fundamental code. It opened up a world of possibilities, hinting at the potential to manipulate genes, to correct errors in the blueprints of life, and perhaps even to enhance our biological inheritance. For decades, this potential remained largely theoretical, constrained by the limitations of available technologies. However, in recent years, a revolution has been brewing in the field of genetic engineering, fueled by the development of powerful new tools for manipulating DNA with unprecedented precision. At the forefront of this revolution is a technology known as CRISPR-Cas9, a revolutionary gene editing tool that has taken the scientific world by storm. CRISPR-Cas9, often shortened to CRISPR, acts like a molecular scalpel, allowing scientists to target specific sequences of DNA with remarkable accuracy. Derived from a bacterial defense mechanism against viruses, CRISPR has been adapted for use in a wide range of organisms, including humans, opening up unprecedented possibilities for genetic manipulation. Imagine being able to correct a single letter misspelling in a vast library of books, or to remove a harmful paragraph with surgical precision. That's the level of control that CRISPR offers, enabling scientists to edit genes, to correct mutations that cause disease, and to introduce new genetic variations with unprecedented ease. The ethics of enhancement, navigating the uncharted waters of genetic engineering. CRISPR and gene editing technologies usher in a new era, fraught with promise and peril. These technologies hold potential for correcting genetic defects, but manipulating the human germline raises ethical questions. Where do we draw the line between therapy and enhancement, and who decides how these technologies are used? The potential benefits are enormous, but so are the risks. The unfolding future of genes. As we conclude our journey into the intimate history of the gene, we stand at the precipice of a new era in genetic science. The tools we have developed, from DNA sequencing to gene editing, have given us unprecedented power to understand and manipulate the very blueprints of life. With this power comes a profound responsibility, a call to wield these tools wisely, ethically, and for the betterment of humanity. The future of genetic science is brimming with possibilities, holding the potential to revolutionize medicine, agriculture, and even our understanding of life itself. Imagine a world where genetic diseases are eradicated, where personalized medicine tailors treatments to our unique genetic makeup, and where we can harness the power of genes to enhance crop yields and feed a growing population. These possibilities are no longer the stuff of science fiction, but are within our grasp. Already, gene therapy trials offer hope for patients with previously untreatable diseases. CRISPR-based diagnostics are revolutionizing disease detection, and gene-edited crops are improving food security in developing countries. The pace of progress in genetic science is staggering, outpacing even our wildest imaginations. As we continue to unravel the mysteries of the genome, we can expect even more groundbreaking discoveries and life-changing applications in the years to come. Our shared genetic destiny. The journey into the gene is not just a scientific one, but also a deeply personal one. It is a journey into our own humanity, a quest to understand the forces that have shaped us, the connections we share with all living things, and the possibilities that lie ahead. Our genes tell a story that stretches back billions of years to the dawn of life on Earth. They are a testament to our interconnectedness, to the shared ancestry we share with all living organisms. Every human being on this planet shares over 99.9% of their DNA, a stark reminder of our common heritage. The differences in our DNA, those minute variations that make us unique, should be celebrated, not feared. They are the source of our individuality, our creativity, and our resilience as a species. As we delve deeper into the genome, we are learning to appreciate the beauty and complexity of human variation. The future of genetic science is not predetermined. It is a future we will write together, guided by our values, our ethics, and our shared humanity. Let us embrace the opportunities and navigate the challenges with wisdom and compassion, ensuring that the legacy of the gene is one of hope, healing, and progress for all.